Okay, guys, today we are going to be talking about translation. Translation is covered in chapter 9 of the textbook, so I highly recommend taking a look at that, especially if you're feeling overwhelmed or confused by the material. There's also a ton of great videos on YouTube. I will upload a couple links of videos I particularly like. I think it's a good idea to look at translation in a video because a lot of times it's hard to kind of imagine these complex molecular processes in our brain without actually like visually seeing it because we are talking about these tiny molecules inside of our cells. And so sometimes it's hard to kind of wrap our heads around that. First, let's go ahead and return to the idea that we have DNA in our cells that goes through a process called replication, but also goes through transcription, which is the process of converting DNA into RNA. Specifically, mRNA or messenger RNA will then be translated into polypeptides or proteins. And so today we're really interested in talking about this translation piece of the central dogma. Some key players are going to be messenger RNA. I just mentioned that's going to be the RNA that will actually be converted into this final polypeptide or protein product at the end. We also have a couple of other RNA molecules, tRNA, rRNA, and then, of course, our ribosomes, which are our machines that do the translation itself. So we'll talk about how these all kind of play a role in translation. First, let's talk a little bit about polypeptides. So when I say the word polypeptide, that usually means, poly meaning many. It's going to be composed of several amino acids, or you could call it an amino acid chain. These polypeptides are assembled at ribosomes, which are organelles that basically all they do is translation. There are 20 different amino acids that can serve as these building blocks for the polypeptide chains. We have what we call covalent bonds forming in between the amino acids, and that is going to ultimately give us that final polypeptide chain, which can then be further folded and processed to be a functional protein. The cool thing about amino acids is they do have these chemical structures that allow them to behave in different hydrophobic or hydrophilic manners. Here is kind of the basic amino acid structure. So all amino acids have this central carbon here in the middle, an amino group, a carboxyl group, and this side chain. So depending on which amino acid we're talking about, the side chains can vary quite dramatically in chemical composition and polarity and things like that. An enzyme in the ribosome will actually catalyze the formation of the peptide bond between the carboxyl group of one and the amino group of the next. So you end up with this long chain. And as I mentioned, this R group is going to be distinct. Some can be electrically charged, some can be polar. One other thing I'll mention right now is that throughout this PowerPoint, I do have this molecular vocab written here in this little light blue box. These are just kind of vocab words I want you to make sure that you are familiar with and you might see appear on future assessments or exams. So here are the amino acids. I do not expect you to memorize these. If you are a biochemistry major, if you're taking biochemistry, you will very likely have to know all 20 of these amino acids, the structures, and their chemical characteristics. What I want to get across here in genetics is simply that there are several different types of amino acids, and they have different chemistries. So we can have nonpolar, we can have polar, we also can have positively and negatively charged side chains. And these are important for various reasons because the structure of these proteins, which are made up of these various amino acids, are doing lots of different jobs in the cell. And so it's kind of cool to think about all of the potential uniqueness each protein can have based on the composition of which amino acids are present. As I mentioned, ribosomes are what we can consider machines. They are going to be containing multiple rRNAs or ribosomal RNAs and proteins that will essentially allow them to be very efficient at translation, or essentially translating mRNA into the proper amino acid polypeptide chain ordered by these specific codons. So we'll talk about the codons. The codons are essentially, if you look at this diagram here, you can see that we have these codons that are going to be in groups of three. So three nucleotides will be a codon, and that's going to be really important for making sure we match the codons appropriately and add the correct amino acids to this growing polypeptide chain. So the mRNA sequence is going to dictate which amino acids get added during translation, and the boundaries of translation are defined by a start codon as well as a stop codon. We also have some untranslated regions. So that's what this UTR is showing here on this diagram. This is going to be areas that are going to be part of the mRNA, but outside of the translated regions that will eventually be polypeptide. So looking at this diagram here, you can really see how as we go through the central dogma, DNA to RNA to polypeptide, we are kind of reducing and specifying the information. So the gene is quite large. As you can see at the top, we have this large gene region. A specific part of the DNA will then be transcribed, and a specific part of the mRNA will then be translated. As you already know, polypeptides do have various levels of organization. So the primary structure is going to be just that chain of amino acids that come out of the ribosome during translation. The secondary structure is going to then be considered either an alpha helix or a beta sheet. So these are just kind of like just a slightly more complex versions of an amino acid chain. We then have tertiary protein structure. So this is going to be where these beta sheets and alpha helixes get folded. And then finally, the quaternary protein structure is going to be multiple of these. So we're getting really complex multi-subunit proteins that are going to be involved in various cellular functions. And it's really cool how you can see how a simple chain of amino acids can really become a very functional, highly specific protein. Ribosomes in both bacteria and eukaryotes perform three primary tasks. They bind mRNA and find the start codon. So we'll talk about what start codon means. This is essentially where translation starts or begins. The ribosomes will start facilitating this complementary base pairing of the mRNA codons and the corresponding tRNA anticodons. So here in this diagram, we have the codons on the mRNA, and then we have these, which are tRNAs that have been charged, meaning that the correct amino acid is bound to them. And they will basically complementary pair the correct codons to add the right amino acid to the chain. So it's going to add the amino acids in the correct order, and it's a very specific code, right? Finally, ribosomes are also responsible for catalyzing the formation of the peptide bonds that we mentioned between the amino acids on this growing polypeptide chain. So we'll talk a little bit more about like the tRNA charging and versus like a charged and an uncharged tRNA molecule. But essentially, the mRNA gets threaded through this ribosome. The codons are going to match up to these tRNA anticodons, and you're going to get this long polypeptide chain come out. Ribosomes are really cool. They are going to be specific with the number and sequence of rRNA molecules and types of proteins. This can vary slightly between different origins of life, so bacteria versus eukaryotes. We'll talk a little bit about the differences between bacteria and eukaryotes, but translation generally is pretty similar. Ribosomes are all going to have two subunits, a small and a large. I feel like I always kind of draw it like this, but there's many different kind of structures of these ribosomes. We're going to consider these ribosomal subunits in Svedberg units, which is, you can just kind of assume it's like this made up unit that's based on size, but also shape and hydration. So It's not necessarily additive, but it is going to give us a general idea of the size of these ribosomes. For bacteria, so E. coli is our classic bacteria that we use as examples a lot of times. We see that the large subunit is around 50S, the small subunit is around 30S. So remember, these S's are Svedberg units, they're based on size, but you can just kind of assume that it's a size unit and that we have a 30 and a 50. As I said, it's not additive, so the fully assembled ribosome is 70. 30 plus 50 is not 70. I realize that might be confusing, but ultimately it is going to give you the estimation of size based also on hydration state and shape. So those are the ribosomes. For E. coli, remember, we have a 30 and 50 adding together to give us a total of 70S ribosome. Eukaryotic ribosomes are a little bit different. We are going to see here for the eukaryotic ones a 60S large subunit, a 40S small subunit, and the fully assembled one being an 80S subunit. As I mentioned, the ribosomes are going to be containing many different types of proteins and rRNA molecules. No need to know all of like the specific sizes and details of this. What I'd really like you to remember is that we do have a large and a small subunit and kind of these general sizes for eukaryotic versus bacterial ribosomes. I'll go ahead and point it out because it is listed here. We have three main sites in ribosomes, E, P, and A. I always remember this because EPA is the Environmental Protection Agency. That's just kind of what's in my head. So you can come up with another way to remember it if you like. But we do have A, P, and E. mRNA actually gets threaded through like right to left, which is kind of backwards when you're used to reading left to right. But we'll talk a little bit more about what each of these sites does. And here they are. So, as I mentioned, we're actually looking at mRNA going through right to left. The A site is the amino acyl site. This is going to bind a new tRNA molecule with that's correctly charged with the right amino acid to be added to the polypeptide chain. Then the peptidyl site, the one in the middle, this P site, will hold a tRNA and attach the polypeptide. So that catalyzation of the new covalent bonds will happen there in the P site. Finally, the E site, the exit site, is going to allow the tRNA to fall off and go away, while basically this large ribosomal subunit is going to contain a channel where you're going to have this growing polypeptide chain coming out. So that's how it works. It's relatively simple. It is a very efficient machine, though, and it does quite well at translating mRNA into polypeptide chains. Here's just a cool picture of the 3D structure of a ribosome. So they're pretty small, 25 nanometers is pretty darn small. And we've been able to image them using some really cool techniques like cryo-electron microscopy. And we've been able to get a lot of information about how they are made up. You can see that it is a combination of proteins and rRNA here. Of course, the tRNA is going to be specifically in this E, P, and A sites. Pretty cool stuff. So, translation is going to happen in three primary phases: initiation, elongation, and termination. The phases are the same in bacteria and eukaryotes, at least considering that they're called initiation, elongation, and termination. There are a few small differences, though. We'll mention a few of them. Basically, how the start codon is identified and the initiation part, but they're all going to be relatively the same in terms of the main stages and how translation occurs overall. So let's first talk about initiation. So initiation is going to happen when the small ribosomal subunit will find this mRNA molecule at the 5' end and specifically find the start codon. So this is the most important part. The start codon is going to be how the ribosome can can basically detect the mRNA and say, okay, time to get translating. We also are going to have the initiator tRNA carrying that first amino acid pairing with the start codon. Once that happens, and this is all happening again on the small ribosomal subunit, then the large subunit will come in to complete the ribosome and allow translation to really begin. So the subunits are really detachable, right? So the small subunit can be floating around, basically looking for mRNAs, finding that start codon, and then allowing for the large ribosomal subunit to come in and complete the ribosome. We also have things called initiation factors. You can also write this as IFs. This is going to help guide the assembly of the ribosome and the binding of the tRNAs. And then GTP is going to be our energy needed for this process. tRNAs are going to carry the amino acids. So when the tRNA has the amino acid, it's considered charged. If there is a tRNA that's empty or doesn't have that amino acid attached, it's considered uncharged. So in bacteria, specifically in E. coli, but you can kind of just assume this is the same for most bacteria, we have six molecular components coming together to initiate this translation. The small subunit, the large subunit, the initiator tRNA, three different initiation factor proteins. So these are just helpful proteins that are going to guide the process. And then our energy source, which is GTP. So for most of initiation, our small 30S subunit is going to be affiliated with this initiation factor, which prevents the subunit from binding until it finds this 5' mRNA and the start codon. That is when the subunits can come together to initiate translation. And I would like you to remember IF3, this is the initiation factor, which is going to be affiliated with a small subunit and prevents premature binding of the two subunits together. The Shine-Dalgarno sequence. So, what happens in bacteria is that this pre-initiation complex will form. We have the 16S rRNA and the Shine-Dalgarno sequence on the mRNA base pair. So, what is this Shine-Dalgarno sequence? It's basically this purine-rich sequence of about 6 nucleotides. It can vary a little bit. And it's going to be 3 to 9 nucleotides upstream of the start codon. So our start codon is here, AUG. That's like our classic start codon we see across all levels of biology. You can think of it, the way I like to remember AUG is that AUG can also stand for August, like the month of August, which is typically when school starts. So you can think of Aug as August, start of school, start codon, where also translation will start. So the Shine-Dalgarno sequence is actually upstream of the start codon. So it's up here before the start codon. Um, And this complementary pyrimidine-rich sequence is found near the 3' end of the 16S rRNA. So the rRNA in the ribosome will actually be able to match up and pair with a Shine-Dalgarno sequence to allow the start codon to be detected. The Shine-Dalgarno sequence, as I mentioned, it can vary between species. So these are all different species of bacteria. You can see that there is this consensus where there is some similarity with all of these different species, right? So the consensus sequence is like the most commonly seen nucleotides at this position. So AGG, AGG. You don't need to remember that, but you can make sure you know what a consensus sequence is and that the Shine-Dalgarno sequence is going to help the ribosomes find that beginning side of the mRNA to initiate translation. And then ultimately, the start codon is where that first amino acid will be paired to the mRNA. Okay, the second step of initiation. So now the Shine-Dalgarno sequence has been recognized. The initiator tRNA will bind to the start codon, where the P site will be once that ribosome comes together. The amino acid on tRNA is modified. So it's actually, and this is a very bacteria specific thing, but we see this informal methionine or F-met being our first amino acid. It's obviously attached to a tRNA. So this charge initiator tRNA is called tRNA, and then you could do a superscript F-met. So F-met is very specific to being that first amino acid for all translation activities. We have another initiation factor, IF2, and a GTP molecule binding to the P site, facilitating binding of tRNA F-met and other initiation factors in order to kind of form this initiation complex. We are still only looking at the small subunit of the ribosome. So the big subunit hasn't even come on yet. We're still in the initiation process where these things are being recognized and that very first charged tRNA is coming in with FMET to start the translation process. So finally, to finish up the initiation process, the big 50S subunit will come in to complete the ribosome. The union of these two subunits is going to cost some energy, so we're going to have GTP to energize this reaction and make sure it's assembled appropriately. We'll have these initiation factors kind of float away or dissociate, and this is going to allow for this full 70S initiation complex to be complete. And we're ready to go. We're ready to start translating. Before we move on to the elongation, I wanted to mention some key differences with eukaryotic translation initiation. So what we were just talking about was bacterial, obviously. Very similar again, we're not talking about anything crazy. The same main things are happening, but there are some key differences I want to highlight. So, the kind of easy thing about translation is a lot of times these initiation factors, and then later we'll see elongation factors and termination factors, these transcription factors that are there to help the process. In eukaryotes, we're just going to add a lowercase e, and that literally just means eukaryotic initiation factor. So eukaryotic initiation factor 1, eukaryotic initiation factor 3. So we're literally just adding an E. So pretty easy to remember the differences. The E is just going to signify that it is found in eukaryotic organism. So these eukaryotic initiation factors will help bind the small subunit, form that pre-initiation complex. It's going to also have this initiator tRNA. In this case, we have methionine still as our first amino acid, but it is not FMET, right? It's not that informal methionine. There's no formal group attached. So that's one really key difference is that in bacteria, it's always FMET, but the first amino acid in eukaryotes is just MET or methionine. The mRNA will be recognized, it will join in this pre-initiation complex, and then we can consider the initiation complex. Once that's formed, it will scan along to find the start codon. And start codon is almost always AUG again. Remember AUG for August, beginning of school. It's also the beginning of the mRNA, the start codon. And that's always going to, of course, bind to the methionine. So once that start codon, AUG, is detected, it can be found also through a consensus sequence. In eukaryotes, we're going to call this the Kozak sequence. So remember in bacteria, it was called the Shine-Dalgarno sequence. Not quite the same, but same concept in terms of it's a sequence that can be recognized in order for translation initiation to occur. So the Kozak sequence, named after its discoverer, Marilyn Kozak, is located nearby the start codon. So this is going to allow the recruitment of the big subunit, the 60S subunit, to the complex. Again, always using GTP for our energy source here. And then once that complex forms, we have the entire ribosome put together. The eukaryotic initiation factors will dissociate or float away. You can see here the Kozak sequence is just going to kind of have the start codon in it. So in this case, it's ACC, AUG, G. And the AUG is our start codon that will pair with tRNA methionine. So, a couple of key differences when we're talking about translation initiation. AUG is usually the start codon in both bacteria and eukaryotes. However, the first amino acid might be different based on just this formal methionine that we find in bacteria specifically. So when we're talking about the initiator tRNA, that first charged tRNA that comes to match up with that start codon, we can write it as tRNA FMET or tRNA MET. And that formal group again is kind of the biggest difference between the initiation of translation in bacteria versus eukaryotes. Okay, let's talk about elongation. So elongation's kind of pretty straightforward. Once initiation has occurred, the mRNA just starts going through and these charged tRNA molecules start coming in, they start getting recruited to the A site, and then the ribosomes will help form these new peptide bonds between the sequential amino acids. The ribosome will move along the mRNA in the 3' direction and just continue adding and adding amino acids to finish up the whole polypeptide translation. And that is what elongation is all about, is simply using this chain reaction to continuously add the correct amino acids. In bacteria, polypeptide elongation is going to be aided by these different elongation factors, or EFs. We also can sometimes have other ribosomal proteins that help, but basically charged tRNAs are going to affiliate with this elongation factor 2 and GTP and then enter into the A site. The tRNA with the correct anticodon sequence will pair with the codon. So it's very important that they're matching appropriately, because if they were not to match appropriately, then you would get the incorrect amino acid added to the polypeptide, and that could potentially be really bad. A lot of times proteins are very sensitive to even a single amino acid change. So it's important that we keep the integrity of the polypeptide chain intact and that we consistently add the correct tRNA molecules to it. So when the tRNA pairs with the mRNA codon, GTP will be used to release this elongation factor 2 from the tRNA. Then a enzyme called peptidyl transferase will catalyze that new peptide bond between the amino acids at the P and A site. So that's happening here. And the polypeptide will continue to elongate and transfer to the tRNA at the A site. So here we have this little spark where the peptide bond formation is happening, and basically the amino acids will just continue being catalyzed and added on to the growing chain. So this enzyme, peptidyl transferase, is an important vocab word to remember. This is the actual enzyme that will allow the new peptide bond uh, formation to occur between the amino acids. Once that happens, the tRNA will then exit through the E site or the exit site, which makes sense. We're still using GTP hydrolysis here. We have elongation factor G translocating the ribosome, moving it 3 nucleotides towards the 3' end of the mRNA. So the ribosome is essentially moving along the mRNA molecule. So it would definitely be helpful to watch a video, because it is kind of cool to watch these ribosomes do their job and to move along the mRNA as this polypeptide chain continues to grow. The tRNA at the A site will then be moved to the P site, and this opens the A site for the next charged tRNA carrying the anticodon. So it's just going to be a continuous process, and they're just going to keep coming and keep getting paired correctly to the codons on the mRNA. In eukaryotes, very similar process for elongation during translation. The only difference is, again, we're just going to add this little e. So instead of elongation factor 2 or elongation factor G, like we see over here in bacteria, the eukaryotic homolog is just going to be EF1 or EF2. So essentially what I want you to remember, you don't have to remember these numbers specifically, but just basically that we can call them elongation factors in bacteria or eukaryotic elongation factors in eukaryotes, which makes sense. It's not that hard to remember. Okay, now let's talk a little bit about translation termination. So this is that third phase of translation where we're going to have to come to the end eventually, right? So elongation is occurring. We're adding amino acid after amino acid, and this cycle will continue until we reach one of these stop codons. So we have our start codon. It makes sense that we would also have a stop codon. Stop codons are usually UAA, UAG, or UGA. I don't have a great way to remember these stop codons. The one I always remember is UGA because that is basically the University of Georgia, and I grew up in Georgia, so UGA was a really big school. If you guys have any tips on how to remember this, please share with me. But these are the possible stop codons that the ribosome might encounter. All organisms will use release factors, so RFs, or eukaryotic release factors. I guess that's not too surprising at this point. Oops. And so these release factors will help bind the stop codon and the A site. When the polypeptide bound to the tRNA at the P site will then be released while the release factor is ejected. And finally, the ribosomal subunits can separate, disassociate, and float away. So essentially, we're just recognizing the stop codon and using these release factors to end the translation process. In bacteria, the release factor RF1 is going to recognize stop codon UAG or UAA, and RF2 will recognize UAA or UGA. Finally, RF3 will recycle RF1. I don't need you guys to know all of these molecular details, but simply that release factors are going to be the ones that are recognizing these stop codons. In eukaryotes, we have termination accomplished by a single release factor, ERF1, which will recognize all three possible stop codons. Eukaryotes also have a second release factor that will recycle ERF1. So everything has a job, everything has a place. It's a very efficient process. And there's all these little, transcription factors through all the stages, initiation, elongation, and termination, to kind of make sure that everything is going well and all the players are where they need to be. So at the end here, we see the ribosomes are disassociating all these release factors and things are kind of going away. And we have our newly formed polypeptide chain. This is just a quick table kind of showing the homologs between bacterial and eukaryotic organisms. So we have RF1, RF2, E, RF1, RF3, E, RF3, and then the ribosome recycling. There's not a homolog in eukaryotes, but again, these are kind of extra details. As long as you know that release factors are involved in translation termination, that's what I care about the most. As I mentioned, translation is fast and efficient. So we just kind of walked through step by step, but this is happening very quickly all the time. It's an ongoing process so that your cells are constantly doing translation all the time for all types of different proteins. So there's speed, there's accuracy, there's efficiency. Cell biologist estimates that each bacterial cell, so this is like an E. coli, has about 20,000 ribosomes, and that makes up about 25% of the mass of the cell. So the cell is really investing in these ribosomes, and they're super important for constantly creating new proteins, which are what is going to allow cellular activity to happen. We've also been able to find these polyribosomes, which are super cool. These are basically structures that have groups of ribosomes that are all translating the same mRNA. So you get these really kind of crazy structures where there are like all these ribosomes on the same mRNA molecule. And you can see they have all these polypeptide chains growing. So as I mentioned, it's fast and efficient, especially if you're going these polyribosome route where you're going to have just constant translation occurring. Here is what the polyribosomes look like in real life. Like here's some actual microscopy pictures of them. And you can see that for a lot of mRNAs, it makes sense that we would want multiple ribosomes translating at the same time because we need a lot of that protein. So it's just going to make everything go much faster. So, in bacteria, one other really big key difference between bacteria and eukaryotes is the coupling of transcription and translation. So, this only happens in bacteria where transcription and translation can happen at the same time. In eukaryotes, we have what we call compartmentalization. This basically means that translation and transcription are happening in two different places in the cell. So in eukaryotes, the cells have nuclei or a nucleus where transcription is happening. So we're getting the mRNAs produced inside. Then they have to be exported into the cytoplasm for translation to occur. So here are our little ribosomes waiting for the mRNA to arrive. In bacteria, there is no nucleus. So transcription and translation can happen simultaneously because there is no compartmentalization. So this is a really, really key difference between bacteria and eukaryotes. So let's talk a little bit about the genetic code that will translate messenger RNA into polypeptides. We obviously have these specific codon sequences, and they get matched to specific amino acids. We have these tRNAs or transfer RNAs that are going to interpret and act upon that information in mRNA with the anti-codon sequences. In the genetic code, we have 64 different codons. But if you remember at the beginning of this lecture, I mentioned we only have 20 possible amino acids. So this leads to 61 codons specifying amino acids, three of which are stop codons. So 61 codons, 20 amino acids. That math is not adding up. What this means is that there is some redundancy in the genetic code. So all amino acids except for methionine and tryptophan are specified by more than one codon, at least two codons. These are what we call synonymous codons. So basically, multiple codons can code for the same amino acid. Synonymous, redundancy, these words are good to know about the translation and the genetic code for translation. So here is a beautiful genetic code wheel. We can call it the wheel of death if you want. You can see that it has a lot of information about how different codons will give you specific amino acids. I like the wheel of death better than the chart on the left, but you can look at it whichever way you want. So starting here in the middle, let's say we have GAG. So we have G, A, G. That is going to give us the amino acid glue. Let's say we have a codon that is CAC. So we'll have C, A, C, and CAC is going to code for his. And then these single letters are kind of the code that are going to correspond to this three-letter code. So obviously we can have histidine, HIS, also coded just by the letter H. So we have 20 amino acids around this big circle, but we have 61, well, 64, including the stop codons, of the possible codons. Now, so we know that there is this redundancy in the genetic code, But something else that's really cool is this third base wobble idea. So tRNA molecules can have different anticodons for the same amino acid. These are considered iso-accepting tRNAs. So even though there's 61 different codons that specify amino acids, most genomes have 30 to 50 different tRNA genes. What this means is that there is this relaxation of the strict complementary base pairing, specifically at the third base of the codon. So this is what we're going to call the third base wobble. So you can see here at this example on the right-hand side, we have the mRNA codon GAC. In this case, we have GAU. The anticodons can match up here, CUG, CUA. And so ultimately, both of these codons will give us a ASP amino acid on the polypeptide chain. And it's because of this flexibility in the third base specifically. So the first two are more strict, right? We have to have a C and a U and a G to A on the mRNA. But the third base has some flexibility. It can be different and ultimately code for the same amino acid. And so just this inherent redundancy in the genetic code allows for this third base wobble to kind of reveal some flexibility. Most of these synonymous codons are going to be grouped into pairs that only differ by the third base. So for example, here we have CUG and CUA. They're differing only by that third base wobble, right? And so they end up carrying the same amino acid, even though they are different, just at that third base pair. This flexible pairing is going to be key. And one thing I will mention though, even though we do have this flexibility at the third base, a pyrimidine must still base pair with a purine. So it's not like anything goes, but it is more flexible than the other two sites. So here are just a few more examples. In this case, we have these three different codons that ultimately are going to code for ILE. And then here we have the two here, which gives us serine. The only part of the codon that's changing is that third base site, right? So the third base wobble is where the flexibility actually exists. So I say it's almost universal genetic code because all organisms, bacteria to humans and everything in between, have a very similar translation process. And because this genetic code is practically universal, we can use things like bacteria cells to produce important proteins from plant and animals. Translation is going to be translation in really any organism, with obviously a few specific differences, but the basic process is the same. Let's talk a little bit more about these tRNA molecules. So we kind of already talked about this, but I want to make sure we have the specifics down. So tRNA molecules are transcribed from tRNA genes. So this is a good example of RNA happening to be the final product. So we have these tRNA genes. This is our DNA. They get transcribed into tRNA, and then that's it. tRNA is not translated. It does have a crucial role for the process of translation, but only mRNA molecules are the ones that will then be translated into polypeptides. The correct charging of each tRNA molecule is crucial. Obviously, that we need to have this integrity. If you have different amino acids getting attached, it can really mess up the protein at the end. We have a couple of important enzymes to remember. So amino acid tRNA synthetase, or you can just call it tRNA synthetase. These are enzymes that will catalyze the addition of the correct amino acid. So I've already mentioned these before, but I just wanted to really highlight how important these enzymes are to basically allow these iso-accepting tRNAs to be recognized. So tRNA synthetase is going to be this large molecule. So in this diagram, it's this thing over here. The acceptor stem of the correct tRNA is going to fit into this active site here of the enzyme. The active site contains the amino acid to be added to the tRNA. We have ATP, another type of energy, providing the energy for the amino acid attachment. So in this particular example, we have glutamate, which is being added to the tRNA stem here. So we have this 3' acceptor stem. This is where our amino acid will be added, and the tRNA synthetase will be the enzyme that's actually charging the tRNA molecules so that now this tRNA can float on over to a ribosome and be connected with the appropriate codon based on its anticodon um, down here at the bottom, and then allow glutamate to be attached to the polypeptide chain. So finally, a lot of things happen after translation too. So once we have successful translation, we simply have a chain of amino acids. The production of functional proteins is not necessarily complete. A lot of times these polypeptides have to be folded or they have to be kind of put together in their functional structures. Typically, this is going to be tertiary or quaternary structures that we talked about at the beginning of this lecture. So this folding and post-translational processing often is going to involve chemical bond formation, sometimes chemical modification and other modifications that will basically allow the proteins to be finalized into their final form and then, of course, transported to their final destination. So this is happening in the cytoplasm, but a lot of times the proteins have to go to specific places, so they then have to be transported. So post-translational polypeptide processing can include various things of this nature. Sometimes we can remove an amino acid or several amino acids from a polypeptide. For example, FMET. Do you guys remember what FMET is? This is that first amino acid found specifically in bacteria. It's going to pair with a start codon. However, FMET is not typically found in functional bacterial proteins. So, a lot of times FMET gets cleaved or basically cut out of the polypeptide once translation has been completed because FMET's not really needed. It just happens to be there because it's always there matching with the start codon. So this post-translational processing is going to just be removing any amino acid from the polypeptide chain that we don't need in the final functional protein. A lot of times we also can have modification of amino acids. The most common one is going to be phosphorylation. Phosphorylation is carried out by enzymes called kinases. So kinases are super common. We find them all across biology. They're going to be enzymes that specifically phosphorylate things. Sometimes phosphorylation can activate, sometimes it can inactivate. It can do various things to proteins. Other enzymes can also modify amino acids in other ways. So instead of adding a phosphoryl group, we can also add a methyl group or a hydroxyl group or an acetyl group. And this kind of changes the chemistry of the proteins and allows them to be specific for different functions in the cell. Sometimes we can also have carbohydrate side chains. So all these different modifications can basically continue to specify the protein and get it to its functional form. Cleavage of polypeptides can also happen. So rather than just cleaving out one single amino acid, like I showed you with the FMET example, sometimes polypeptides can actually be cleaved into multiple segments. So there are certain segments that might need to go different places or do different things. A really good example of this is the hormone insulin. So, insulin is first produced as something we call pre-proinsulin. So, that's what you see up here at the top. We have a couple of different chains. So, we have chain A and chain B. And so, this has to be cleaved to be fully processed. Once we cleave these pre-amino acids, so we can cut off the first few or whatever we need to cut off here, we then get something called proinsulin. Proinsulin will then have the chains. We have these disulfide bonds forming, and then ultimately, these chains also get cleaved. And so, insulin, the final hormone that we're trying to get to, insulin, is actually going to be kind of multiple segments of this original polypeptide that have been cleaved and then bound together in different ways. So here are just a few additional examples of different post-translational modifications. We can have hydroxylation, ubiquitination, lipidation, etc., etc. All these different things are going to add functionality to proteins and allow them to do the jobs that they are intended to do. Another thing to mention is that we have endoplasmic reticulum that often is involved in protein sorting. So proteins that are destined to remain in the cell are typically translated at these like free ribosomes that are kind of floating around the cytoplasm. But if you guys remember back to like probably high school biology, you probably remember the rough endoplasmic reticulum where ribosomes are kind of studded onto this organelle. Polypeptides that are destined for secretion, leaving the cell, are a lot of times produced at the rough ER. This allows for the transport to happen more efficiently once the proteins are made. So here is a good cartoon of kind of what's going on at the cellular level. So here we have the rough ER. The mRNAs are coming in and the ribosomes are translating them into polypeptides. They then get packaged into these vesicles. They will go through the Golgi apparatus and then eventually they will get secreted from the cell. Pretty cool stuff. So, we've been talking about these codons, these anticodons, and this is basically a triplet genetic code. And this was found back in 1961. They did some cool experiments where they were able to kind of look at how changes in the reading frame can really alter final protein. So when I say reading frame, what that refers to is the specific codon sequence as determined by the start codon. So we're always going to have that start codon, AUG, right? Here we have it here. This is going to make sure that the correct reading frame is going to be used. If you were starting like one nucleotide off. So here's our start codon here, AUG, but in this example, it's starting at CAU. Well, that's giving us a completely different polypeptide chain. Now, you might have a couple that are going to be correct, but overall, you're going to get a completely different protein. And so it's very important that for translation across all organisms, that we use the start codon to allow us to use the correct reading frame. Frame shift mutations are going to alter the reading frame, which makes sense. Like the word kind of explains what it is. It's shifting the frame, and it's going to really mess up anything downstream of this mutation. So in this made up example, our wild type is going to say, you may now sip the tea. We have these triplet codons that are going to specify these specific words. Now, if we had some kind of addition or insertion, we have this C being inserted here, it's going to completely throw off anything downstream. So everything gets shifted. This is a frame shift mutation. And now we get these nonsense words that don't make any sense. You can imagine that in an actual cell, this would be just coding for various different amino acids, giving us a different polypeptide chain at the end. Same thing would happen if you had a deletion of some kind. So here We had the deletion of the letter W. This is also going to cause a frame shift mutation and shift everything downstream. Sometimes we can have frame shift mutations that get restored. So sometimes a second mutation in a different location can happen. So in this case, we have our mutant addition. So the C got inserted, messed up all the downstream codons. But then we had a reversion mutant deletion. So something got added, then something got deleted, and this actually restored the frame shift, the reading frame. And so we still have, something messed up here. Maybe a couple of amino acids are not right, but everything downstream is then going to be corrected. So that is possible to have these reversions. A lot of times it just kind of depends on the protein, how important the certain amino acids are to the protein's function can kind of dictate how severe a mutation will be. So it's important to realize that most scientists agree these genetic codes are read continuously. There's no gaps, there's no spaces, there's no pauses. So if there were gaps or spaces, then an insertion here, well, I guess it's actually the T is the insertion. It would only affect this amino acid, but we know that is not the case. Instead, an insertion or deletion will shift the reading frame and affect the entire polypeptide downstream. This is a nice summary of translation. So I just kind of highlighted some of the key differences between bacteria and eukaryotes. The overall translation process is very similar. The main differences, in my opinion, are the sizes of the ribosomes. Eukaryotes have slightly larger ribosomes and subunits. This initiator tRNA, so we have F-met for bacteria and just regular met for eukaryotes. The initiation site, so we have a Shine-Dalgarno sequence in bacteria, Kozak sequence in eukaryotes. A lot of these transcription factors, whether it's the initiation factor, elongation factor, or release factors are going to be very similar, with the difference literally just being their names. We often call the eukaryotic ones eukaryotic elongation factor, eukaryotic initiation factor. But all of these factors are kind of helping the process, making sure the key players are going to the correct place. In both bacteria and eukaryotes, we have this third base wobble. We have tRNA synthetase that will charge the tRNAs with the correct amino acids. And then, of course, the frame shift mutations can happen in both bacteria and eukaryotes. So this is kind of a nice summary table. Again, I do recommend watching videos. If you're a visual person, it's kind of nice to see translation actually happening. What I would like you to do is complete assignment number 6. This is an assignment that I would have preferred us do together in class in small groups, but it will still be a very useful assignment. So you can work in groups if you'd like, or you can work independently. It's up to you. This assignment is going to really kind of drive home some of the major processes in the central dogma. So transcription, translation, post-translational processing, splicing, things like that. So please look at this assignment on Canvas. It will be due on Friday. You may want to use this. This is basically the wheel of death, like I call it, where you can identify what each codon will code for each amino acid specifically. Please remember the rules of the different base pairings. So CGs, As, and Ts are used depending on RNA or DNA. Make sure that you feel comfortable with translation. Chapter 9 has a lot of great information. There's also great resources online. I'll link some videos. The next time we meet, we'll be talking about Chapter 10 or chromosomal abnormalities. So that should be super interesting. Please make sure you complete your assignment number 6 and upload it by Friday at 2:15, which is when our class normally would end. Again, we're not meeting, but I do expect you to have a good understanding of translation and complete this assignment. I hope you all have a wonderful spring break. Thank you for being flexible and allowing me to do this asynchronous week. And please email me if you have any questions. Thank you.